All right, it is 12:30, so we will get started for today. And I just wanted to start with this article that came out yesterday in Translational Psychiatry. Um, in it, the researchers, what they did is that they had rats and they fed some rats uh, high sugar diets when they were very young. And what they have found is that these rats that get the high sugar diets, their gut bacteria, the microbiome actually changes compared to the controlled rats. And these rats have lower cognitive functions than normal rats. So they're showing that there's a linkage between high sugar diets when you're young, which leads to a different gut microbiome, which leads to lower cognitive abilities. If you'd like to read more about that, this is actually an open source journal. So feel free to just look up for this title. Figured it was interesting since we have been talking about metabolism this whole semester here. So just to remember what we are talking about on Tuesday, we have been looking at the organs now with metabolism, how everything is interconnected. We're kind of taking the step back from where we were, this whole semester looking at each process. Now we're looking at how all of the organs were involved. In the very last slide, we were talking about the signal transduction pathways, where a signal can come and interact with the cell, and from there lead to a big change in response. And it's usually due to three main pathways. Shown here, the RAS pathway, the receptor tyrosine kinase pathway, or the CAMP, adenylate cyclase pathway. And we're just going to talk a little bit more about this until we get on to the lecture that I made for today. And one thing we need to talk about is AMP-dependent protein kinase, or just AMPK. AMPK is a very essential kinase when it comes to balancing metabolism. Because as we've been seeing this semester, the amount of ATP, or rather the amount of AMP to ATP in your cells, control a lot of different things. So your cells don't actually know what levels of ATP you have. There's no like ATP sensor anywhere. What we have is an AMP sensor. And the idea is, as you burn ATP, you make AMP. And from there, that's when your body knows how to either start breaking down things to make energy or store stuff. Store your energy for future uses. It's all about the AMP levels. And because of that, different enzymes will be activated or inhibited by AMP. And one enzyme that controls a lot of that is AMP-dependent protein kinase, AMPK. So when AMPK is active, what it will do is that it will go and activate the pathways that break down metabolites, which makes sense because if you have AMP around, that means you need energy. AMP will also, or AMPK rather, will also go and inhibit the pathways that build things. Again, makes sense. If you're low energy, don't build things. So this graph in the bottom left shows you everything that AMP, well, not everything, but it shows you an example of the type of things that AMPK is responsible for controlling. So in the heart, what AMPK will do is do glucose to lactate, right? Take all your glucose and start making it into lactate to get that energy. In the liver, so the red lines means it inhibits this. It inhibits gluconeogenesis, so don't make glucose. It inhibits lipogenesis, don't make fatty acids. It inhibits cholesterol making, so don't make cholesterol anymore because you're low energy, but it will promote the breakdown of fatty acid to energy, right? And in fat, it will break down the creation of fatty acid from glucose. But interestingly enough, what AMPK does is it will stop your triacylglycerides from breaking down into single fatty acids. So even though you need energy, AMPK is going to say don't break down your energy stores for fat. Keep it in the triacylglycerol form. Can anyone think of a reason why AMPK might do this thing? It sounds counterintuitive. Well, it is counterintuitive. It just sounds wrong though, right? If you're low in energy, you would want fatty acids in your blood. Yet our body's designed not to release fatty acids. Can anyone think of a reason why that is? It's not obvious, I'll say that. I forgot to plug in my board. One sec. Let me mute this so you don't hear me crashing stuff. All right. So the reason that people think that even though AMPK is activated by levels of AMP, right? That turns it on. But it stops the breakdown of triacylglycerols and the fatty acids. The reason people think this might be is because fatty acid inside of your bloodstream, if you have too much of it, that can be toxic, right? Fatty acids, nonpolar, and we need special proteins to carry them. So maybe, you know, if this was unchecked, your fat, your adipose tissue, all of your adipose tissue, right, would release triacyl or fatty acid at the same time, maybe clog your arteries, clog your veins super fast. Or, you know, you could have a buildup like we see in high fat diets where you have cholesterol buildup. So that could be a possible reason why, even though fatty acids have a lot of energy, we don't actually just release them from triacylglycerols even with AMPK. So that's the idea behind there. And that's the hypothesis at this point. But any questions about AMPK, what is its role, you know, what processes its controls, or anything like that? All right. And I actually have a big picture thinking question. Again, isn't obvious, but let's see if you can piece this together. So this question is, physical inactivity might lead to insulin resistance. On a molecular level, how might this happen? So taking what we've talked about today and what we talked about in previous classes, see if you can map out a pathway logically of, okay, if I'm not active physically, what does that mean? And then why could that lead to an insulin resistance, excuse me. So I'll give you all, a couple minutes. Just think about that. See what ideas you can start to percolate. Again, this is not one of those obvious ones. So try to put the pieces together. If you're not working out or being physical, you'll always be in a high energy mode. Yeah, that part is correct. And I see you're still working on it. But yeah, so let me just fill out that piece of the puzzle. So if you're inactive, your ATP levels will always be high, right? Because you're not burning it. Therefore, your AMP to ATP ratio will be low. You won't be making AMP, right? Because you're not burning ATP. So continue on from there. What effect could that have? So having low AMP, what does that affect? So you're in high energy mode. So CMP is a signal for low energy. CMP won't be utilized. High ATP will signal for storing to degradation. Low AMP will inhibit biosynthesis. All correct. Yes, very good. But let's connect that with what we talked about on Tuesday, right? So what this all means is if you're high ATP, what won't you release? Well, you won't release insulin all that often. Because you're always in like that constant, oh, I'm fed. So I'm not going to create it all that much. But the main thing is though, so this is, it's a little bit, but not that much. The main thing is you're no longer going to have AMPK. AMPK won't become active all that much. And one thing that AMPK does is similar to insulin in that it helps to increase the amount of GLUT4 in your cells, in the activity of GLUT4. And remind yourself, what does GLUT4 do again? And how is GLUT4 activated? Or, yeah, activated. So GLUT4 is what we talked about on Tuesday. It's a glucose transporter. And it's activated by insulin in your muscles, right? Yes, insulin sensitive glucose transporter. That's correct. So the idea is if AMPK doesn't get turned on, then the amount of GLUT4 in your cells will decrease, because AMPK is one of the things that help regulate the amount of GLUT4 inside your cells, and it helps regulate their ability to work. So even if you have insulin around, so let's say you produce insulin, you don't have as much GLUT4 around now because you're never in that low energy state. So your cells aren't gonna produce GLUT4, and so you're gonna start becoming insulin resistant a little bit. Your cells just won't, your muscle cells just won't, recruit the GLUT4 to the plasma membrane anymore, because you just don't have much of it to begin with, because your body's never telling itself to make it, because you're never in low energy. So that's kind of the whole string of why you need to do exercise and why inactivity could lead to some insulin resistance there. So questions about that idea? I guess in a nutshell, if I was going to explain it in like a sentence, not only does activity go down, the amount can also go down. And so that's why it's harder. That's why it seems like your cells aren't responding to insulin. You just don't have the GLUT4. They're not responding as well. So that's another important role of AMPK there. Helps to activate GLUT4. So let's look at some other hormones here. So here's adiponectin, and this is one of the hormones that also controls AMPK, which we just saw is super important for everything. And this molecule has 3 different stable conformations: low molecular weight, medium molecular weight, and high molecular weight, right? LMW, MMW, HMW. And really, LMW is a trimer, right? So you have 3 copies. This is a hexamer, and this is just a lot, or multimer. And what adiponectin does is that it'll go and bind to specific receptors on your cells, so your liver and your muscle cells. And when they bind, they will phosphorylate AMPK, or they'll start the cascade to phosphorylate AMPK, which we just talked about means you're going to start breaking down things to make energy. So this will inhibit gluconeogenesis, stimulate that fatty acid oxidation, glucose uptake, and increases how well your insulin works, right? So this will make sure that like when you get fed and you release that insulin, your body really responds to it. And a decrease in this, therefore, is again associated with insulin resistance, just like how we looked at a lack of exercise can do it. If you don't have adiponectin, then you could start becoming insulin resistant because your body just doesn't respond to the insulin as well as it should. So that's one protein, one hormone. Well, it's a protein and a hormone, but that really regulates metabolism for us. And it's all through, again, turning on AMPK. That's what this protein does. And another protein that's always fun to look at studies of is leptin. Leptin is the full hormone. You release this when you're no longer hungry. And what we see in this picture of the mice, if you create a mouse that doesn't have leptin, they get super fat because they never know that they're full. They just keep eating, right? They never get the signal, oh, my stomach's full. For humans, the amount of leptin inside of our bodies increases with the percentage of body fat that we have, because leptin is made by your adipocytes. Therefore, more adipocytes, more leptin. However, this is actually bad because as we create more leptin, we just kind of ignore the signals from leptin, right? Your body just gets used to it and doesn't respond to it anymore. You become tolerant to it. And this is one way that, you know, we can see obesity. It's just you overeat, and continual overeating, your body will just stop responding to the I'm full signal and just say, well, okay, I guess we're not full because we're still eating, so just keep on eating. And the biochemical reason of why maybe we're resistant is that, you know, either the receptors just are always bound, so there's no more like on-off switch. They're just always on until they're being ignored, or just the amount of receptors are decreasing. Now, it is possible to be born without leptin. It's rare, but it is possible. And so those individuals are usually obese because, again, they don't get the signal to say, I'm full, right? And, you know, when I was in grad school, the person who had the biochemistry department at Iowa, he studied leptin and metabolism and all that. And his anecdote about leptin was like, in animals, it seems to work a lot better because like dogs, well, it depends on the dogs, but dogs sometimes will just stop eating if there's food still there because their leptin's working great. Humans, we seem to consciously ignore the leptin signal if I'm full and keep trying to eat. Again, I guess that depends on the dog, because I've definitely seen my dog eat too much and then not have a good stomach afterwards. But yeah, that's the full hormone, leptin. So any questions up to this point about anything we've talked about? Right. Last hormone is neuropeptide Y. And when leptin stops affecting us, that usually leads to higher concentrations of this peptide called neuropeptide Y, or NPY, which is released by your hypothalamus. And what NPY does is that it makes you hungry and leads to you storing fat. Usually, leptin and insulin will block NPY from working because they do opposite jobs, right? Leptin is I'm full, and insulin is you have energy. NPY is I'm hungry, I need to store energy. because who knows what I'm going to eat again. Store that as fat as the best way we can carry it. So if you don't have leptin anymore, based on what we just talked about, therefore there's nothing inhibiting NPY. And so it's just kind of like cascading on itself. You overeat, you overeat, you overeat. You no longer are sensitive to leptin. Instead, your brain is saying, okay, here's this other chemical that says you're always hungry, you're always hungry, you're always hungry. Store that fat, create more fat. And it's just an ongoing cycle. Another enzyme, ghrelin, that's actually released by your stomach when it's empty, which will go in a boost NPY. So don't have anything in your stomach, ghrelin will go and make sure NPY is released to say, oh, I'm hungry. And you have something else that inhibits NPY called PYY3 through 36. Horrible name. So yeah, whoever found that was not good at naming things. But that is just a peptide that will stop NPY, saying, oh, we're no longer hungry now. And what NPY are, not NPYs, but what happens sometimes in humans, and especially babies, is that we can go and deal with our excess energy through thermogenesis. So that's what that bullet point, diet-induced thermogenesis, means. That means it is possible that even though we're storing up all this energy, we don't actually need to, because we can just burn it as heat. This is what is called brown fat. So that's how babies like stay warm. They have a good amount of brown fat, where the ATP that they create is strict, or it's not even ATP, the proton pumps, they're used to directly generate heat. As you get older, unfortunately, you lose that brown fat for the most part. But it has been found that adults can still do thermogenesis in their white fat. And that type of fat's called beige fat, half white, half brown, beige, I guess. That's white fat that seems to be able to do thermogenesis. And the way that this works is that you have UCP1, uncoupling protein 1. And instead of the proton gradient going through ATP synthase, you go through UCP1, and all that energy of movement is just used for heat. That's it. And people have found a peptide that will increase UCP1 production. And actually, when you exercise, your muscles release this peptide. Urisin, I want to say that's how that's pronounced. Urisin, probably. So that shows you how exercise itself can be used to burn fat. It's not only the process of using more calories by like lifting heavy stuff or running, is that it might actually turn on and create UCP1. So excess energy is just burned away through heat. So it is possible that an anti-obesity drug could be made from this hormone, because if you just give it to people, they turn on UCP1. But that hasn't been tested, and it could be dangerous because you could mess with people's ATP synthase and accidentally, worst case scenario, kill them, but lead to a lot of other problems. So I'm guessing that's probably why it hasn't been tested, but it is something that is plausible at least. And that should be it for our PowerPoint that we started on Tuesday. So up to this point, any questions about anything? Is there a possibility to use PP or PYY336 in place of leptin, or no, because they are strictly due to a location where they are secreted? So are you talking about like if someone's leptin resistant, maybe try and do PYY336 and see if that would like restore the or stop NPY. I would guess So the way this things usually are in biochemistry, and I don't know these pathways like super intimately that I could tell you, okay, and our PYY interacts with this and interacts with this and interacts with this, and therefore that's why MPY stops. So I'm going to say that probably off the top of my head here, PYY will definitely go on a different pathway than leptin. Their mechanisms for actions are probably different. So you couldn't do a one-to-one substitution, I would think, of PYY doing the same job as leptin. But saying that, I don't actually know 100%. That would just be my gut feeling on it, based on how pathways usually work. And leptin, like we're just going on very, very, very like surface level of these proteins. As we've seen when we've gone into different pathways, once you like dig deep into it, there are a lot of moving pieces. And I haven't studied all those moving pieces, right? So I can't give you a sufficient answer on that. My gut would say probably not, but I actually don't know. So it would be more of a side interaction. To really answer that, I would have to look at the actual pathways that PYY and leptin travel through. What do they interact with to make their mechanism stop? And like you said, what other things are going on? What other pathways do they affect to have their overall impact? Because it's easy to say, like, as we're learning this in like biochemistry class, we are like reducing a protein down to like 2 sentences that has been covered in probably like, hundreds of pages of academic work, right? When we just boiled that down to 2 sentences. So it's easy to say that, oh, leptin doesn't or makes you full, but like understanding how everything interworks, that would take more study. So that's why I can't give you a nice and clean answer, but it is a fun thing to think about and a fun thing to research if you have the, if you would like to do that. And as always, when something like this comes up, if you wanted to look into the possibility of that and see what's been done on that and teach me, like send me some links and like write me a half a paragraph on like, how are PYY and leptin different, or how is there anything that's looking into leptin resistance with PYY? And if you find anything, and send me like a half a page, just like, here's what I found, here are the references. I'm always happy to give extra credit for that. So that type of stuff, since it's actually, biochemistry related, is yeah, if you want to do some digging on that and let me know what you find, that would be great. But yeah, unfortunately, I know a lot more about the physics of biochemistry than super in-depth pathway of everything. But good question, good thought-provoking question. All right, so with that, let's switch PowerPoints here. And our next topic is we're basically going to talk about what happens when you're in different metabolic states. By that, I mean, we're going to start with starvation. What happens when you don't eat? How does metabolism change? Well, to really understand this, we have to keep in mind that even when you don't eat, the blood glucose levels in your blood are nearly constant. Remember, we talked about on Tuesday, your brain needs glucose. If it doesn't get glucose, you die. If it dips below the amount it needs, you go into a coma and eventually die. So it's very important your blood glucose levels stay constant. Unfortunately, in your body, you have less than a day's worth of carbohydrates stored up in glycogen. We don't have that much glycogen. Even worse, if you eat a lot of amino acids, we don't store amino acids. We can turn them into something else, like glycogen and triacylglycerides, and have it stored as that, but we don't have like a ready supply of amino acids. We either use it to build or we'll change it into something else. So no one-to-one storage for amino acids. So let's go over the way that people starve most often, and that is after you wake up, right? So after you wake up, what has happened? Well, since you haven't eaten in a long time, you increase the amount of glucagon flowing through your system because you haven't replenished your glucose. And you don't have insulin really going through your system as well because you haven't ate. Therefore, fatty acids will start to release from adipose tissue, right? We're going to start releasing energy now. Also, your muscles will stop increasing uptake of glucose because of our friend GLUT4. Remember, GLUT4 is turned on by insulin. Insulin goes down, muscles not going to increase glucose. And that's so your brain can still use glucose. So your muscles sacrifice themselves. But muscles will use the fatty acids that have been released from your adipose tissues as fuel instead. Over here on the right is just how much energy a typical man at 70 kilograms. What is that? I'm not too good at kilograms to pounds myself. So let me get my handy dandy calculator out. 70 kg to lbs. So this is a 154-pound individual. They have roughly 166,000 calories stored up. See, most of it is fat. If you broke down all your protein, your muscles, you'd have a good amount. Your glycogen is tiny, 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 tiny. And then fuels that are just in your bloodstream as well. Yeah, 150, very good. I can never remember kg to lb like in my head. So let's look at how fast you lose that glycogen. So after not eating, your liver will just chop up your glycogen to make glucose because glucagon is the one that directs that, if you remember back from February when we talked about that. And we also increase gluconeogenesis. So any energy that we, or any metabolites that we have laying around, we're going to try and make them into glucose because the brain needs glucose. So after 40 hours, 96% of the glucose in your blood after 40 hours of not eating comes from the liver. This chart just shows you how fast the amount of glycogen in your liver goes down. So this is time from eating, glycogen in millimolar. After 40 hours, you're basically done. All these different lines, I believe, are just different trials, different samples. So it decreases really fast. Your kidneys are also one of the organs that will make glucose in this process. We did look at how your kidneys do take up amino acids. So they can do gluconeogenesis from amino acids. Unfortunately, we cannot make glucose from fatty acids. We can get energy from that, but your brain can't get glucose from fatty acids. And the reason is you can't really make pyruvate to oxaloacetate easily from acetyl-CoA. So, and I had that little, explain this statement. I already answered the first one. What hormone is being released to promote gluconeogenesis? That's glucagon. But what does it mean? What does that sentence actually mean? And like, if you were to explain it to someone not in biochemistry, why can't pyruvate oxaloacetate easily be formed in a net manner? What does that mean? take that scientific sounding sentence and say it in your own words. That's the exercise. Can you translate that for me? So I'm not sure if I can translate it easier. I probably could, but let me explain it with all the tools that we know. All right. So acetyl-CoA is made from pyruvate. We looked at that. I memorize because delta G for oxaloacetate is so high. That is of the citric acid cycle. Yeah, the delta G of the first step of the citric acid cycle is extremely high. And that does feed into it, yes. So one way to make acetyl-CoA is through pyruvate, and pyruvate can make glucose, right? We looked at that. Fatty acids, when you break it down to acetyl-CoA, right? So this is the theoretical pathway. Now, pyruvate to acetyl-CoA is irreversible. And that's done by that pyruvate carboxylase complex we talked about. That's removing a CO2. And it has those 5 different steps in it, if you remember that. So we cannot go back from that. Therefore, the only way we can make glucose, if we start with acetoacetate, is that we need to make oxaloacetate. Because oxaloacetate, if you remember, we can make that into phosphoenolpyruvate and go to glucose. But the only way to do that is the citric acid cycle, which you lose 2 carbon dioxides, and you also have to use oxaloacetate in that cycle. So you're just going to be wasting a lot of carbons if you try and change acetylacetate to glucose. So that really doesn't happen. Once you're acetyl-CoA, you're not going to make that into glucose. We break down fatty acids into acetyl-CoA, so we can't use that for glucose, which is very unfortunate. But you can use glycerol from triacylglycerols to do that. So glycerol can be made from glucose. So indirectly, we can use our fats. Proteins can. And so one of the big things is that your muscles will start to break down and you will make glucose from that. And 1 second, let me go quiet that dog because it's hard for me to think when a dog's barking. All right. So after a few days of not eating, you are going to start making ketone bodies because your brain can use ketone bodies as well as glucose. So as we just saw, we cannot take fatty acids and make glucose. You can make fatty acids and ketone bodies. So that's how we're going to stay fed during many days of starvation. Just to show you how your brain adapts, after 3 days, of not eating, 33% of the energy of your brain is gotten through ketone bodies. After a month, it's going to be 70%. And as we saw on the previous slide, that one way to make glucose is to just break down your muscles, and those can go directly to make glucose. Amino acids can. But as your fasting increases, the rate of muscle breakdown will slow. Why would that make sense? Why is that a good thing? As the longer you don't eat, the slower you degrade your own muscles for energy. Why did we evolve like that? Why is it a good thing that we did? Third threshold to maintain our mobility. Yep, that's exactly right. Because if you broke down all your muscles and then you couldn't move, well, you're definitely not finding food again. That's for sure. So the idea there is slow down the amount your muscles break down so you can still move and find food. That's kind of the reasoning why we have that safety switch. So how fast you can survive from not eating is based on how much fat you have instead of how much muscle you have, because you just stop using muscle after a while. And if you're a highly obese individual, and this was actually done as shown in the picture down there, and I think I went over this in the video as well, you can live over a year without eating. So this is Angus Barberry, 456 pounds, went to a hospital, and they did a clinically supervised weight reduction program where he didn't eat for 382 days. They gave him like vitamin supplements, and he dropped down to 180. His body just used all the excess fat as energy. And there didn't seem to be anything wrong with him. I think he lived to like 60 or 70. So it seems completely possible as long as you get vitamins and minerals to do that. I am no way endorsing that because that would have to be done under the strict supervision of doctors. I think he went there like all the time just so doctors can continue monitoring all his levels. But in practice, your body can just switch over to all your energy storages for your ATP needs. And as we talked about, there is some proof out there in mice and rats that reducing the amount you eat on a daily basis may increase your lifespan due to reactive oxygen species. Again, that's not really shown, hasn't been shown in humans just because we didn't know about it. And so maybe check back in a couple of 300, 400 years when people have actually tried this to see if it actually works in humans. But yeah, that's just an interesting fact about prolonged fast and how your body can just adapt and use all the energy we carry around. Let's get to another type of situation where your metabolism is not in the correct order, and that is diabetes, which is the third leading cause of death in the US. And two types: either you are not creating insulin, which is type 1, or your cells are no longer reacting to insulin. That's type 2. And your cells are going to starve because glucose import is going to be impaired. And why is that again? Why do we need insulin for our cells? Well, not all our cells, some of our cells to intake glucose. It's our friend GLUT4, right? Insulin. We'll go and turn on GLUT4. So it all comes back to GLUT4 eventually. That's just one of the ways, at least. I guess I didn't need to delete that. So what happens when your body stops importing glucose, or your cells do? Well, your AMP levels are going to rise because you're not doing glycolysis anymore. That means all your signals for low energy will start to turn on. AMPK will become activated. You're going to start breaking down your triacylglycerols. You're going to start doing fatty acid oxidation. You're going to start making glucose. You're going to start making ketone bodies. It's going to be similar to if you were fasting, because your body doesn't know you have energy. And what happens is that you keep making ketone bodies at such a high rate that your blood starts to get a little acidified, acidosis. Your pH drops. Your blood buffer can no longer handle the amount of ketone bodies. And your urine becomes acidic. And you'll start breathing out acetone. As we talked about when we talked about the ketone bodies, one way to know if a person has diabetes and they're not treated is that their breath smells like nail polish remover. It smells like it is acetone. So if you go into the lab, there's those bottles of acetone around that if you accidentally put on your skin, it feels really cold because it evaporates super fast. That's the stuff you're breathing out because of the ketone bodies' spontaneous decarboxylation into acetone. Like I said, there's two types. Now I have the breakdowns here of like type 1 versus type 2. So let's start over type 1. And usually type 1 is you're born with it. It's because the beta cells in your pancreas are either not there or they don't work. And it's the beta cells that are sensing the amount of glucose in your blood at all times and releasing insulin to tell your body if you have high levels of glucose. And usually it's autoimmune, where, you know, early on in your development or as like a teenager, that's why it's called juvenile onset diabetes. You just create an antibody that targets your pancreatic beta cells and you accidentally destroy them. So you need constant insulin injections and a balanced diet and a way to externally measure your blood glucose levels. And you're basically doing the job of your pancreas then. And individuals type 1, usually lifespans are reduced by a third just because there's so many possible complications. Kidneys, nerves, cardiovascular, everything's going to be possible to go wrong if your blood glucose levels spike or dip. Right? And what can happen is if you have too much glucose is you get hyperglycemia. That can lead to blindness. That's what the image is showing down here. Basically, the lens, your lens proteins just get covered in glucose, and so they no longer function. Possible way to alleviate this is that if you get a transplant of new pancreatic beta cells, because that's basically the only thing that's wrong. So if you can get those beta cells either transplanted or somehow working again, that should alleviate type 1 diabetes. However, the majority of cases that we see now is type 2. So 90% of all diabetes is type 2 and may affect up to 10% of all adults. And this is when you're insulin resistant. You can make insulin, and a lot of times you make too much insulin, but your cells just don't respond to it anymore, which means that your glucose in your blood will never be used up, right? Your cells aren't importing it, so you get high levels of glucose in your blood. And so your beta cells, your pancreatic beta cells, are always getting the signal, hey, we have very high blood glucose levels here. Release insulin. And then you just keep flooding your body with insulin, causing this problem to snowball. Well, the more insulin you release, the cells are just going to ignore that signal because they keep getting bombed with it. So they're not going to respond to it. And all this is very complex, but it does seem that if you eat too much constantly, your cells just stop making insulin receptors, so they will just shut it off. Also, if you have a lot of fatty acids in your blood, this also prevents insulin from doing its job correctly. And there are some drugs out there that will help to promote glucose going into your muscles and stop the amount of glucose being released by your liver as a way to control those blood glucose levels. And by doing that, you can increase the amount of AMPK inside your cells that are active, that can, which are active under like a low energy. So that's actually what happens during type 2, is because your body doesn't know that you're fat again, and AMPK is just not going to turn on, as we already talked about at the very beginning today, of AMPK's job of I'm low energy, so I'm going to turn on and start breaking down energy or breaking down metabolites. So that's like the difference between type 1 and type 2 is just type 1 don't make insulin. Type 2, you don't recognize you're making insulin. On the bottom, this chart here, this is for type 1 diabetics, and that's just the blood glucose level, right? And these spikes, breakfast, lunch, and dinner, and time in minutes. So you can see a normal person, they roughly stick around 5 millimolar. If you ever get a physical where they take your blood, they're going to ask you not to eat for so many hours to get that baseline here. If you're diabetic, you can see that it's just already incredibly high, and how much more it spikes when you eat, right? So for a normal person, it goes from 5 to 7, 5 to 7, 5 to 7. If you are type 1, you're starting at a baseline of 12, and you're going to 17 because your cells just aren't absorbing it. And that's where these problems come in. And then it takes a long time for this to go down on its own. So that's diabetes. And any questions about that before we move on to our next topic about metabolism not working correctly? All right. So how else could metabolism not work in your body right? Well, cancer. Cancer metabolism is quite different than normal metabolism. One, cancer cells take up a lot of glucose. So one thing cancer does in your body is that they just steal glucose from the rest of your cells. They'll do this with and without oxygen, right? They'll just constantly intake glucose. And the reason why they can take in all this glucose and use it is because a lot of the times they overexpress PFK2, and this will increase PFK1. Remember, PFK1 is the enzyme that is the main control point for glycolysis, phosphofructokinase, and it's controlled by the amount of fructose bisphosphatase 2 and phosphofructokinase 2, right? It's controlled by those levels. And so one way that cancer cells can get around that is if they just modify the DNA and say, well, don't turn this off, just keep PFK1 on. So that's one way we can just continuously cycle through all this glucose. And as you make a lot of glucose, You're going to be making a lot of lactate as well. And when you make lactate, that actually promotes vasodilation, which makes sense, right? If you're making a lot of lactate, that means you don't have oxygen because you're not doing the citric acid cycle. So in response to a lactate buildup, your cells will naturally try and recruit blood vessels to get oxygen delivered to it. Well, cancer cells, they kind of use that as a way for more and more and more blood vessels going to them, which means they get more glucose. And how do they modify DNA? It's through this molecule 2-hydroxyglutarate. And 2-hydroxyglutarate can go and interact with the enzymes that modify DNA, which means that these changes in the enzyme amounts inside your cells, they can make that permanent. Normally, everything is fine-tuned. Everything can be switched on and off. Cancer cells can just say, you know what? I really want to do glycolysis. How about we keep on PFK1 forever? That would be great. So let's just go modify the DNA and do that. And I will say, like, even though I'm talking about cancer cells as like they know what they're doing, they don't. It's all due to random chance and mutations. And basically it's evolution at that point, where like one cancer cell might accidentally always turn on PFK1. And so that one's just going to grow out of control, right? And as they grow, more and more mutations happen. And then maybe one daughter cell can outcompete all the other cells. And so they'll grow and grow and grow. Eventually, one daughter cell will learn how to migrate throughout the body, and then they'll grow in different parts. So yeah, it's all due to random chance at that point, whether you get the mutation or not. Here on this chart, the arrows are showing you what parts of the main metabolism paths cancer cells like to do. The green arrows means you have upregulation. The beige arrows are basically downregulation. So in general, cancer cells don't care about making ATP all that much, which, you know, it kind of makes sense, because if they're getting a lot of glucose, well, just make it into lactate and you'll get all the energy you need, because you're just keep importing glucose. And instead of going through this citric acid cycle, a lot of that glucose is going to go through pentose phosphate pathway. So you can make nucleotides, or you're going to make proteins, or you're going to make fatty acids. Any amino acids that we get through the bloodstream as well, we can also just say, let's make those into nucleotides and proteins as well. So we are not going the energy pathways we have mainly focused on for cancer cells. And I do have a question, why would a cancer cell use these pathways over other pathways? So I'll just go ahead and answer that. It's because you're growing so fast, you just need to keep building stuff, right? The regulation for the cell cycle is turned off in cancer cells. So they need to keep growing, split, grow, split, because there's no stop signal. One of the hallmark mutations of a cancer cell that spreads rapidly is that they just turn off all the checkpoints that usually tells them stop reproducing. You are done at that point in your life. They turn that off, and so they need to keep making stuff. So that is one of the big differences we see in cancer cells versus healthy cells, the amount of glucose they use and what they use that glucose for. So questions about that idea? All right. So at this point, even though I don't have a slide here, we are leaving behind metabolism. We are in a new chapter. So all the pathways, all that stuff, for the time being, depending on how the rest of the semester go, we might go back to it for some things, but this is like a hard cutoff right here. So we're going to move into a brand new, different field of biochemistry now. And that is DNA, how DNA is made, how DNA is repaired and regulated. So a tangent to metabolism maybe, because DNA controls everything. But just so you're aware, just get your minds reset of, okay, that was the metabolism 3 month section. Here's the DNA for the last month section. So just in the last few minutes, let's just go over the very basics of DNA synthesis that hopefully you've learned in previous classes before. So DNA, the replication of DNA is carried out by DNA polymerase. And for that, we need single-stranded DNA as our template, ssDNA. This only happens to the 3' ends of DNA through a nucleophilic attack, as shown here on the bottom. Here's our 3' hydroxyl. Here's our new nucleotide to be added. It's a DNTP. DNTP. DNA nucleotide triphosphate, so 3 phosphates. When we add the new nucleotide, pyrophosphate, that's what that's called, is removed. And we get our new DNA with a phosphate holding them together. Now, just about every single DNA polymerase scientists have found will only add nucleotides to the 3' hydroxyl. There's a handful that can do it 5' as well, but the majority do it at 3'. Therefore, that's why we say DNA is replicated 5' to 3'. We always add to the 3' direction. We can never go backwards. Most part. We'll look at some enzymes that can actually go backwards. So if we're looking at circular DNA, as in E. coli, what happens during synthesis is that we form these theta structures. You'll see little replication forks, and it kind of looks like a bubble or an eye is forming, because in E. coli, since it's a circle, it's relatively easy. You get one polymerase working this way. You get the other polymerase working in the opposite direction. And they go around until they hit each other and fall off. And while they're replicating, it looks like a little eye is forming. Right? And so the branch point where this is happening is called the replication fork. That's a term we'll be talking about a lot. So anytime you hear replication fork, it's just where the polymerase is working currently. And replication is what's called semi-discontinuous. And I actually have a question, what's that term called? Well, it's right there. So we have 2 different strands, the leading strand and the lagging strand. So the leading strand works where the DNA is the parent. So here blue is parent. So the parent is 3' to 5', and then we can add 5' to 3' continuously. The semi-discontinuous part is called 5'. It's the 5' to 3' end, because we are going backwards now, because we can only add in one direction. And so this is where the semi-discontinuous part comes, because we have to do this in different fragments. And they're named after the person who discovered these Okazaki fragments. And so the question, why do we have to do this? Again, this is a question for like when you're going back to these slides and reviewing, make sure you like answer this yourself. It's because we can only add DNA 5' to 3'. And so for the lagging strand, you have to do that in segments. You can't do continuous 5' to 3' because you're continually unzipping new DNA where you can start, okay, now I can start a new 5' here and move until I hit the back of that strand. These Ozaki fragments are small. Eukaryotes 100 to 200. Prokaryotes are 1,000 to 2,000. And the way that they work is that you actually use RNA to start. So for a polymerase to work, for DNA polymerase to work, you need a template there to begin with. You need like a staging platform. You need nucleotides already. You can't, for most DNA polymerases, you just can't start adding DNA to nothing. You need what's called a primer. That primer is going to be made out of RNA because the enzyme primase can add RNA de novo. You don't need anything there to begin with. You can just start adding nucleotides. So primase adds a primer that's 1 to 60 nucleotides long, depending on the organism. Then polymerase, as shown here, will extend from this. As more and more DNA is unzipped, we create a new primer. DNA polymerase will add, and we also have a different polymerase that will chop off the old RNA and replace it with DNA. And the fragments are going to be combined by an enzyme called ligase. And since we're out of time here, I will just answer these questions. But again, good review. If we are adding 2 DNA fragments together, what is the amino acid that is critical for this function, or rather what type? By that, I mean like positive, negative, polar, and nonpolar. Remember, DNA is incredibly negatively charged because it has a lot of phosphates. So we would need a positive amino acid. And we actually did look at this in biochemistry 1 for like 4 minutes on one slide. So it's okay if you don't remember what it is. Lysine is the important amino acid in our ligase. So DNA ligase connects DNAs together thanks to enzymatically important lysine. All right. So next week, we're going to continue our journey down DNA synthesis. And we're going to move into what happens when your DNA is injured. What are the mechanisms that we have to employ to repair like double-strand breaks and single-strand breaks? So that's what the future has for us. But for right now, we are out of time. So I appreciate you coming, joining me today, watching me on YouTube. Always appreciated. If you have questions about anything, always please email me. I'll put up some homework problems, so just as a way to refresh what we went over and to give you points for studying, forcing you to study. Otherwise, I have nothing left for you. So I hope you have a great weekend, a good start of your April, and hopefully I will see you soon. Take care, everybody.